Can everyone see the slides? Make sure yes, I'm not
Yes.
muted this time. So
you're good.
All right. So we have today a slate of very dynamic and interesting speakers as opposed to just me and Rick. But before we get to that, I'm going to talk a little bit more broadly about global proteomics. So we touched on this throughout the talks yesterday, because it's essentially what we do most of the time is look for patterns of changes in complex samples. So the overall goal here, it may seem odd to trot out a definition of proteomics on the second day of the workshop, but this kind of broadly illustrates the challenge that we face here with what we're trying to do. So we can have in a biological organism a variety of different types of cells, for example, in different phases of the cell cycle under different conditions. And this all changes dynamically over time. So that makes the proteome much more complex and variable than a genome for any given organism. So that's one of the source of a lot of the major challenges that we have to deal with in proteomics, and why a lot of times trying to use mass spectrometry to do proteomics is more complicated and can be more challenging than using gene arrays to measure gene expression directly. So these next two slides show one of my favorite examples of the complexity inherent in what we're trying to do. So even if we're studying a small organism, or not even really an organism, like a virus with 15 proteins synthesized from 9 open reading frames, as with the HIV genome shown here, the biological problem is really going to be a lot more complicated. So if you were looking for, if you're interested in the life cycle of the virus inside the host, then you're really looking at more of an interaction network than a series of independent measurements necessarily. So this is so the red spheres on this diagram here are the 15 viral proteins, and then the smaller yellow ones are the host proteins, and the nodes between them show the known interactions. So a lot of times this kind of picture is what we're trying to capture with proteomics. So you can, it's obviously much more dynamic than you can see in any given snapshot. But this is why the data analysis is so complicated. This is why we need to spend a lot of time and effort and use expensive equipment to make really sensitive, really rapid measurements on a large scale, because we're trying to get at what's changing over time in this kind of situation with with a viral infection or anything that alters the protein complement of the cell from one condition to the next, or over time. So that's why it's difficult, because it's very complicated. So more specifically, some of the challenges we have to overcome are listed here. So most of the time, this is increasingly less true because mass spectrometers are just so much more sensitive now than they used to be. But the general rule has typically always been that we never have enough sample. You can only biopsy so much tissue or collect so much blood, and the protein yield is limited by that. So you have to you have to plan carefully what measurements you want to make based on the amount of sample you have available. So that's one problem. And then, since you're dealing with complex samples, it's a challenge to detect, to make measurements of single proteins and single peptides in that context. So we've got to be able to do all of this, whatever it is we're going to do, we've got to be able to do it rapidly, because we're trying to collect a large amount of information. So if it takes days or weeks to collect data on a single sample, then that just doesn't work very well. We've got to be able to do all of this reliably, as well as quantitatively and in a statistically valid way. So that's, this kind of sums up what, a lot of what we talked about yesterday in terms of the more specific approaches we take to doing this. this kind of sets the stage for what Stephanie's going to talk about with the challenges of and importance of data analysis and doing it correctly. So I'll just, in the next few minutes here, I'll talk a little bit more specifically about a couple of these challenges and how we overcome them. So something we haven't really touched on much, I don't believe, is this the dynamic range issue. So this is pretty much true of it's an issue, at least to some degree, with any complex sample, any cell lysate or anything like that. But it's particularly true of serum samples, of the protein complement of blood. So that's the example we're going to use here. So dynamic range is just that's the difference between the concentration of the most abundant protein in the sample and the least abundant protein in the sample. And with blood, with serum, that dynamic range is around 10 orders of magnitude or something along those lines. So it's a striking example here. So if you take undepleted serum, fully half of it is a single protein, albumin, and the top 10 or 12 most abundant proteins take up 90% or more of the total protein bulk. And then, of course, the precious biomarker you're looking for that's going to be the indicator of disease or something that you can latch onto as a potential therapeutic lead is always going to be in this yellow segment of the pie chart here, which is the pool containing low abundant proteins. So proteomics is not just challenging because you have to detect low-level proteins. It's more challenging because you have to detect low-level proteins in the presence of far more abundant proteins, which is a much more difficult problem. So there are a couple of solutions to that. One is to build really good mass spectrometers that can scan really fast, have really high sensitivity, and so you can get past read past the more abundant peptides and still collect data on the lower ones. But something we still pretty much always do is some kind of separation or fractionation of the sample to deal with this dynamic range problem. So maybe in a few years, we'll be able to lyse a cell or take an undepleted serum sample and just do a single mass spec run on that and identify 10 or 15,000 proteins. But we're not quite there yet in most cases. We can, with a cell lysate, we can get 2,000 or 3,000 proteins, but we can't get really deep into the proteome without fractionating or enriching the sample to deal with this dynamic range problem. So the way you do that with serum is just, you can buy kits these days, antibody-based kits that will bind out the more abundant proteins, the top 12 or 14 now. So that's what we routinely do with serum. Just get rid of everything that you don't care about that's going to get in the way of measuring whatever it is that you are interested in the sample. And that same strategy in different forms is applied to most samples that we analyze. So with cell lysates, we typically don't discard anything, but we fractionate many times the total peptide complement. And we'll look in more detail at the peptide fractionation here in a minute. But other examples would be if you're interested in mitochondrial proteins, it's usually a good idea to take the time and the trouble to do a mitochondrial prep rather than just lysing the entire cell and trying to sort out just the mitochondrial proteins in the end, because you'll get much better data if you can separate out what you need from what you don't need, or at least fractionate the sample so that you can look at it in smaller, less complex chunks rather than all at once. So that's one important way that we deal with complexity in proteomics in a lot of cases. So with the data independent methods that we've seen a little bit of, that's becoming less of a problem. So in that case, we're able to use sophisticated data analysis and database preparation to get around the need in a lot of cases to fractionate the samples so extensively. So yeah, so this is recap from yesterday. So once you get the sample, you fractionate, separate, deplete, whatever it is, then it goes through this whole LC-MS-based system to identify proteins and peptides. So we went all through that yesterday in the with the protein and peptide ID presentation. So real world, how you might, there are a couple of ways you might separate samples other than the serum example. So we don't do nearly as much gel-based analysis as we used to, but this is an intuitive way to think about it here. So instead of doing a single shot analysis of the cell lysate or a pull down experiment in this case, if you can, we used to do this commonly. We just take the sample, the complex sample, run it out on an SDS page and grid it into 24 slices from the top of the lane to the bottom, and then separately analyze each section of the lane. So that way you get 24 times as much instrument time out of it as just running it all together, and you separate, you lower the complexity in each fraction of the sample. So what we're doing here basically is a two-dimensional separation. Dimension 1 in the standard SDS page, and dimension 2 in the on the inline LC column that's attached to the on the HPLC attached to the mass spectrometer. So if you doing that type of analysis with the gel-based approach, you can get upwards of 5,000 or 6,000 proteins under ideal conditions from a single sample. So that's what we're showing here. And then the other the way what we're commonly doing now is skipping the gel and doing something similar with offline peptide fractionation by reverse phase chromatography at basic pH. And we talked about this quite a bit more yesterday, so I won't spend as much time on it today. But I am going to just real quick talk a little bit more about exactly how it works and why it's effective. So the pH here is the important factor. So this offline separation, it's very similar superficially to the separation we do at the LC-MS step. Both are reverse phase separations. So we're doing a two-dimensional separation using reverse phase chromatography in both cases, and the main factor that we're altering is the pH. So this offline version of it we do at pH 10, and the online version, the inline version, right before the mass spec analysis, we do at acidic pH, and that of course has to be acidic to provide protons for peptide ionization. But offline, we can lower we can increase the pH and use basic conditions. So this figure here illustrates why that works. So this shows you on the x-axis retention time for each of these peptides under acidic conditions, and then the y-axis is retention time for the same peptide under basic conditions. So if we just look at one of these, let's take this labeled one, the HK-IPIK. It's got under acidic conditions a retention time of less than 10 minutes, but under basic conditions, under retention time on the same column of nearly 25 minutes. So that means you can get an orthogonal separation for a complex peptide sample by just changing the pH in your two dimensions of separation. Now, if this graph were a straight line from the origin, up along the diagonal here, then it wouldn't work at all. It might be more closer to what you would intuitively expect. But just changing the pH for the reverse phase separation changes the interaction of most peptides enough that you can get a good separation. So I won't go into detail here. So this is just illustrating what we were talking about yesterday, is that we can do this in a strategic way to get a separation and recombine individual fractions into super fractions that will distribute across all of them, that the mass spec instrument time as evenly as possible. So this illustrates again how this works. So if you do two reverse phase separations at the same pH under acidic conditions, This is, panel B here shows what you get. And so the second dimension, you don't get much additional separation. Everything clusters together, which is not surprising at all because you're just doing the same thing twice. If you change the pH so that you're doing a basic separation first and then acidic separation second, you get a much broader distribution here, and your instrument time is filled up nicely, and the peptides are spread out across that the gradient fairly evenly. And then the ultimate result of that is this figure, which I showed yesterday. So you can do this kind of situation with 13 hours of instrument time, occupied fairly evenly across the board with peptide analysis if you do the separation strategically. That's the secret, really basic, I guess, to global proteomics is to start with complex samples and treat them in a strategic way to get the most out of the instrument time you have available applied to the across the complexity of the sample. And then this is a different graph than we showed yesterday, but it's a similar point. So just showing that as you increase instrument hours devoted to a project, you increase proteins identified and quantified, but with diminishing returns. So it takes a lot of instrument time to get out to 10,000 protein IDs, not so many to get to up to five or six, as far up as 5,000 or 6,000. So that's the in a nutshell how we approach global proteomics. So we can stop here for a few questions on this, or anything anyone thought of from yesterday that you want to revisit before we go on.
Any questions?